Ah yes. The tree of life. We're familiar with it. The plants. The animals. The fungi. The trees. To all the fish swimming. In all the 7 seas. We're familiar with our brothers and sisters in nature. But where did we all come from? Who was the first? Introducing L.U.C.A, the last universal common ancestor. So what do we know about him? Well, first, we know he was a prokaryote. Prokaryotes are quite simple, as they are single-celled organisms that don't have a membrane-bound nucleus. Chemolithic autotrophic prokaryotes are able to obtain energy by oxidizing inorganic carbon, which helps them stay alive in extreme conditions. We know that the first life on the big tree, L.U.C.A, was likely a chemolithic autotrophic prokaryote and a thermophile. So what is a thermophile? Well, it means he liked it hot. Particularly, where the temperature was between 60 and 108 degrees Celsius. He always loved to go to the extreme and take advantage of his 3D amino acid structure that twists and folds in response to heat, unlike his XGF, whose enzymes unfolded and no longer worked. Rest in peace. As much as he loved deep-sea vent beach, he could also be found vacationing and visiting his family in heated habitats, like this hydrothermal vent system, terrestrial hot springs, or volcanic islands. And of shallow water environments. Oxygenic photosynthesis became a main form of converting sunlight into chemical energy on Earth. This was a new alternative to the chemical energy conversion that organisms were already doing. Photosynthesis allowed for the rise of atmospheric oxygen, creating a new path in evolution: eukaryotes. And as more and more organisms used this energy conversion process, it led to Earth's oxygenation. So how do we know that L.U.C.A was the last universal common ancestor? Our best evidence is the 16S rRNA. Like DNA, RNA is responsible for the coding of proteins, which are responsible for the function of our cells. RNA is older than DNA, so we can use it to go further back in the tree of life. Why 16S RNA? 16S RNA is special because it codes for a ribosome. Ribosomes are ideal to look at because they contain regions of highly conserved and variable components across species. Essentially, what we're able to do is look at 16S RNA coding sequences of any organism, from a small bacterium like L.U.C.A to a large and complex organism like you or me. So how did we get to this image of the tree of life? It all started with looking at RNA. There are 3 types of RNA that each have their own function in cells. There's mRNA, tRNA, and rRNA. So where do we start? Well, Carl Woese started that process for us. He was the first to start analyzing sequences of rRNA from prokaryotes and eukaryotes. He was able to discover that life forms who shared common sections of rRNA could form groups. We can then take those smaller groups and form them into even larger ones. This was how the domain of archaea was discovered. Because certain sections of its rRNA did not match up with prokaryotes or eukaryotes, creating its very own branch in the tree of life. Scientists were able to find that rRNA, more specifically the 16S rRNA sequence, was found in all living cells, the site of translation for all proteins from mRNA3, and was composed of both conserved and variable regions across species. We were able to expand on the work of Carl Woese by analyzing the 16R RNA sequence of all species to create what we now know as the tree of life. So, how did we go from L.U.C.A to the world as we know it today? That might be cause for another video.